Hi, good afternoon, everyone, and welcome to today's briefing, What is Precision Medicine, with Dr. Harold Varmus. Before Dr. Varmus is introduced, I would like to introduce Congressman Brian Higgins, who represents New York's 26th Congressional District, which includes portions of Erie and Niagara counties. He serves on the House Committee on Foreign Affairs and the House Committee on Homeland Security. Congressman Higgins is an avid supporter of biomedical research. In January, he reintroduced the Accelerating Biomedical Research Act with Rosa DeLauro and Congressman Peter King. This legislation will allow Congress to appropriate additional funding to NIH above the Institute's current cap. If passed, the Accelerating Biomedical Research Act would allow for more than $50 billion additional dollars to the NIH over the next 6 years. Congressman Higgins is also a steadfast supporter for cancer research. He has led efforts among his colleagues to increase funding for cancer research and is engaged with the cancer community on innovations that will alleviate suffering due to cancer and ultimately find a cure. He will shortly reintroduce the Cancer Drug Coverage Parity Act. Insurance coverage has not kept pace with innovation in medicine and the growing trends towards orally and other patient-administered chemotherapy. This legislation allows for oral chemotherapy treatment to be covered by insurance the same way as intravenous chemotherapy treatment. Congressman Higgins' district is home to Roswell Park Cancer Institute, an NCI-designated cancer center. With that, I am pleased to introduce Dr. Higgins.
Thank you. Good afternoon. It's afternoon, 10 after 12. Welcome to today's presentation on precision medicine. I'd like to thank the Congressional Biomedical Research Caucus and its co-chairs, Steve Stivers, Joe Barton, Jackie Spear, and Steve Cohen, for inviting me here today. Precision medicine is an approach to disease treatment and prevention that takes into account variables in genes, environment, lifestyle in each patient. The President's budget proposes $215 million for a precision medicine initiative. This is a start, but we need Congress to increase our overall investment in research institutions. To that end, my colleagues, as has been mentioned, Rosa DeLauro, Peter King, and I have introduced legislation to exempt the National Institutes of Health from sequestration cuts. Our bill would result in a $50 billion increase over what is budgeted for the National Institutes of Health over the next 6 years. With the budget debate about to heat up in Congress, it is my hope that medical research funding will be an area of bipartisan agreement as it once was and as it should be again. Dr. Harold Varmus was awarded the Nobel Prize for his studies in the genetic basis of cancer. He has served as director of the National Institutes of Health and chief executive officer of the Memorial Sloan Kettering Cancer Center. And most impressive of all, he was once the scientific advisor to this very caucus. Since 2010, his leadership at the National Cancer Institute has been outstanding. He oversaw the establishment of 2 new centers, one for global health and the other for cancer genomics. He has worked hard to improve the grant-making process at a time when, due to lack of funding, fewer research proposals are funded than ever before. I've been proud to work with him on behalf of Roswell Park Cancer Institute in Buffalo and on behalf of cancer researchers across this nation. Today, Dr. Varmus is here to discuss precision medicine, what it is, why it presents an exceptional opportunity. I know I speak on behalf of everyone here when I say thank you to Dr. Varmus for his outstanding leadership in public service. Now please join me in welcoming Dr. Harold Varmus.
Representative Higgins, thank you very much. Very nice working with you over the last few years, and I appreciate the efforts that you and Representative DeLauro and King are making to improve our fortunes. This is in many ways a bittersweet moment for me to appear before this caucus. Not only was I an advisor to the caucus for many years in the early 90s, but I was one of the founders with the advice of Representative Peter Gekas, who had been the advisor to a group of scientists who were trying to get other scientists more engaged in the political process. And on October 3rd, 1990, nearly 25 years ago, I appeared at what is arguably the first caucus meeting, along with Lee Rosenberg, who was then the dean of the Yale Medical School. Corey Bargman, who was then a postdoctoral fellow at MIT, is now one of the leading neuroscientists in the country. And we were addressing a funding shortage nowhere near as profound as the one we're currently funding, but one that was severe enough for all of us to be concerned about the future of biomedical research in this country. Almost exactly 10 years later, after I had been the director of NIH, I appeared again at a caucus meeting, this one held in the Cannon caucus room, to talk about the remarkable success we were having as a result of efforts to double the NIH budget over 5 years. Very, very different times, but one in which we argued that the scientists who were engaged in biomedical research still needed to do a better job of reaching out to members of Congress and to make sure that our science is conducted in the best possible way. Now, about 15 years later, here I am again. Hope you're not bored with me, but most of you are new to the process. And the situation has, as you've just heard from Mr. Higgins, turned sour fiscally. My motto at the NCI has been, we still have a lot of money. We still have $5 billion, and we've got to spend that very responsibly while also hoping for increases. In raw facts, we're $177 million, or we receive $177 million less this year than we did the year I arrived, which is not a particularly good track record from my point of view. We're about 25% down in corrected dollars, inflation-adjusted dollars, from where we were in 2003 when the doubling of the NIH budget concluded. So we have a financial challenge, but $5 billion is a lot of money, and my job has been to try to use the money well. And I believe that the rise of this initiative called precision medicine, based on decades of work by the NCI and by the rest of NIH, is emblematic of the fact that we are continuing to make progress, take on new things in addition to the things that Mr. Higgins kindly alluded to. The other aspect of this that makes it bittersweet is that I think some of you probably know, I announced a couple of weeks ago that I'll be leaving the NCI to go back to New York City, where I've officially lived and where my wife still lives, to take up a couple of new positions at the New York Genome Center and at Weill Cornell Medical School. And that will happen on April 1st, no days off. So today, what I'd like to do is give you my own sense of what the precision medicine initiative, at least the part that NCI is playing a major role in, is all about. Why it's now possible, what the problems ahead are going to be, what the NCI is trying to do with the money the President's asking for, and we hope the Congress does provide. Let me start by acknowledging the important role that the President's playing and speaking to this issue as he did on January 30th in the East Room, where I and many of my colleagues were privileged to hear him talk. And I will let you hear him talk very briefly.
The budget I sent this Congress on Monday will include a new precision medicine initiative that brings America closer to curing diseases like cancer and diabetes and gives all of us access potentially to the personalized information that we need to keep ourselves and our families healthier. You can match a blood transfusion to a blood type. That was an important discovery. What if matching a cancer cure to our genetic code was just as easy, just as standard? What if figuring out the right dose of medicine was as simple as taking our temperature? And that's the promise of precision medicine, delivering the right treatments at the right time, every time, to the right person. First, we're going to work with the National Cancer Institute. We want to find the genetic factors that can lead to cancer. And we want to use that knowledge to develop new and more effective approaches to help people beat this disease. Second, we're going to work with the FDA to develop new approaches for evaluating next-generation genetic tests. The way we approve a new gene sequencing technology is going to be different than the way we approve a new pacemaker or prosthetic device. We need to make sure that our approach reflects the differences in technology. Third, we're going to work with the National Institutes of Health to create a research group of 1 million volunteers. And just like analyzing our DNA teaches us more about who we are than ever before, analyzing data from one of the largest research populations ever assembled will teach us more about the connections between us than ever before.
So the President's brought up issues that I will be addressing as we go along, but let me first give you an overview of this notion of precision medicine, one that's been in existence for a long time, but was fairly carefully defined and well studied by a group of scientists working for the Institute of Medicine about 4 years ago. And their report towards precision medicine summarizes things quite nicely. We're used to the idea that many of us do biomedical research. That research gets stored in some form in journals or in databases, and that eventually it begins to influence the way we practice medicine. But what this new push indicates is the possibility of using a much more detailed set of markers for the behavior of disease, integrating that information in a much more effective way through modern computational science, using that to do what is fundamental to precision medicine, changing the taxonomy of disease so that we have new disease categories that give people a better idea of what kind of therapy should be used, what the likely outcome might be, how we might more effectively prevent disease, and the new use of electronic devices for monitoring what physicians do, what they measure, how patients respond, should allow the incorporation of many observational studies into a database that constitutes a knowledge network for different kinds of disease. My own definition of precision medicine emphasizes this notion that what our precise knowledge of disease implies is an ability to make new diagnostic categories using many kinds of information, genetic, other molecular features of disease, as well as factors about etiology that Mr. Higgins referred to. That includes inheriting traits, exposures to environmental causes and behaviors, and using all that information to then approach categories of disease with more powerful tools to both treat and prevent disease. Now, this general notion is not totally new. Over 65 years ago, Linus Pauling first elucidated the precise nature of an anemia. This anemia found particularly in Africans and African Americans, called sickle cell anemia, and he showed that disease was due to a very particular mutation that changes the nature of the hemoglobin molecule. And he made a prophetic but perhaps mistimed statement about we're entering a new era of medicine as a result of this kind of information that tells us why someone's anemic based, in this case, on an inherited mutation. Now, many years later, we still lack good treatment. So this is a sobering reminder that making advances in this molecular and diagnostic range is not always the immediate precursor to solutions. Cancer is the starting point of the initiative the President has announced, and it's worth asking why. And in brief, it's because we have assembled, as I will show you in just a moment, an enormous amount of new information about the genetic basis of cancer. Cancer is at its root a disease of the genome, of the collection of our chromosomes, affecting both the DNA and the proteins that regulate that DNA. And in addition to having all that information, cancer is a useful starting point because it is such a common cause of disease, number 2 in the US, and very high as a cause of death in almost all countries, accounting for about 12% of deaths worldwide. We also have some new drugs. I'll illustrate some of those in a moment, and we have new ways to use the immune system to treat cancers, and I'll mention those too. We've had some amazing success on some kinds of cancer and very slow progress on many others. That's a very important issue. But it does emphasize a very, very important point, that there is not going to be one single cure for cancer. There are lots of different kinds of cancer, just judged by the sites at which those tumors arise. And within any single site at which tumors occur frequently, there are many different kinds of cancer based on the genetic criteria we'll be talking about. But as a result of the tools that are now available, rapid sequencing of genomes, much better analysis of how cells work, new forms of computational devices, the opportunities for improving the situation are enormous. I do want to make a very critical contrast here. When people talk about genetic disease, they frequently mean a disease you inherit from your parents, or even a disease that occurs by a mutation occurring during development. But unlike such diseases, exemplified by sickle cell, which I already mentioned, or cystic fibrosis, cancers are not the products of a single inherited mutation. They're the products of many mutations, and most of those mutations occur after birth, commonly during development and early and adult life. There are some contributions from the germline. We don't know all of those, but we know many of them. But the consequence of this multiplicity of mutations that affect the development of a cancer means that cancers are highly varied, and I will show you some illustrations of how varied they are. And in this constellation of mutations, there's some that are much more important than others. Some that clearly are driving the carcinogenic or the cancer-making process, and others that are simply along for the ride, occurring in a cell that becomes a cancer cell, but not having any functional significance in the formation of the cancer. In fact, cancers, like species, are continually evolving, and that evolutionary process of random change and selection for a favorable trait is a very important attribute that makes this approach to cancer through precision medicine quite difficult, and I will show you some examples of that as well. So we already have a good reason to be optimistic about this kind of approach because we have several remarkable successes. The poster child for this is the treatment of a disease called chronic myeloid leukemia. The most common adult leukemia, about 6,000 patients acquire this disease per year, similar incidence worldwide. Many years ago, when I was learning medicine, almost every patient who developed this disease, usually in midlife, around the age of 50, died within about 3 to 5 years. And in 1960, quite a long time ago, it was discerned by people looking at cell chromosomes in Philadelphia that there was one abnormal chromosome which turned out to have a stereotypic appearance, and this appearance reflected the fusion of two genes, one of which has cancer-causing properties when it's rearranged in a way that activates its enzymatic activity, the enzymatic activity of its protein product. It was then discovered about almost 20 years later that a drug that was being developed for a totally different purpose inhibited that rearranged enzyme. And the result is the identification of a drug known as IMATINIB, but commercially sold and known to most of you as GLEEVEC, which showed remarkable, extraordinary activity even in phase 1 drug testing, and then was very rapidly approved by the FDA. That drug, GLEEVEC, is now known to inhibit 3 enzymes of a very similar type called tyrosine kinases, and as a result is active in several kinds of tumors, most of which are relatively rare, but it's really an extraordinary tribute to the basic notion of precision medicine that this drug and some other drugs that have now been put on the market and are very similar restores a normal life expectancy to a patient who's diagnosed with CML. So that's totally remarkable, but you shouldn't be misled by this because in most cases, cancers prove not to be that simple. Many more mutations that are found in this disease, many of them being innocent bystanders, many different kinds of genes affected as opposed to the virtually monotonous appearance of the translocated chromosome, the Philadelphia chromosome seen in chronic myeloid leukemia. Many more ongoing mutations, although those are also seen in myeloid leukemia, and acquired resistance, which I will emphasize in a few moments, to the drugs that are targeted to specific mutations. Now, we do have drug resistance to GLEEVEC occasionally, but we understand the resistance very well, and we have second-line and third-line drugs that can be used to treat patients who develop GLEEVEC resistance. Now, I've shown you one kind of mutation, a translocation that joins 2 genes and makes one cancer-causing gene active. But there are many kinds of mutations. Commonly, mutations that just change one of the letters in the genetic code and change the nature of proteins made by that gene. Changes in the copy number of a gene. Usually we have 2 copies of every gene. And for many cancer-causing events, I wonder if you could just slow down on the pictures. A couple of they're pretty stereotypic, and 1 or 2 is enough. The changes in number of copies of genes, deletions of some genes, amplifications of others, also contribute to making a tumor. So these things all have to be followed, and to try to pull together a catalog of all these changes and the many, many different kinds of cancer we already know exist because they arise in different cell types. The NIH was able to establish a massive program called the Cancer Genome Atlas, the NCI working together with the Human Genome Research Institute. And over the course of about 7 years, aided dramatically by Recovery Act money that was put into our budget with the strong leadership of Arlen Specter, this program selected initially 3 particularly important cancers to look at, later expanded the list to nearly 30 cancers, and developed the techniques for developing, for acquiring many different kinds of data, both a reading of the sequence of building blocks or letters in the genetic code, but also counting copy number, looking at the frequency with which genes are expressed to make proteins, and looking at many other biochemical properties of chromosomes. And that had to be done with huge teams of people that acquired the samples, analyzed them, computed the results to give us meaningful information for interpretation. And those teams, highly self-sacrificing, funded with the hundreds of millions of dollars every year by our 2 institutes, allowed us to assemble a tremendous amount of information that is, some sense, visually represented here, where across the bottom you have the names of many different tumor types, several kinds of lung cancers, brain cancers, leukemias, and so forth. And on the right, the number of cases that have been analyzed. You can see that several hundred tumors, tumor genomes, and corresponding normal genomes in the same patient have been looked at. Many papers have been published, analysis underway for many of the others. Well, what have we learned so far? Well, we've learned that genomes in cancer cells are altered in very complex ways, that tumors are of the same type under the microscope are very different when we examine them genetically. Even within a single tumor, as I'll emphasize in a moment, tumor cells look different to a certain extent. But nevertheless, this information allows us to define those important diagnostic subtypes. So lung cancers become many different kinds. Glioblastomas of the brain become at least 3 or 4 different subtypes, and so forth. We also find there are many kinds of genomic change, and those changes affect the way different components of the cell oversee the activities of a cell growth, cell death, cell invasion, and other factors that play a role in cancer. Furthermore, cancer genomes have what we call mutational signatures. If we look at the mutations, they reflect exposures to environmental agents, and those can be very revealing with respect to what's happening as the environment predisposes, or behaviors predispose to certain kinds of cancers. And then all of this information is now guiding virtually every aspect of cancer research. It guides the way we do clinical trials, as I'll show you in a minute, and it guides the way we are beginning to administer and will in the future universally, I believe, guide patient care. Some of the issues can be seen by thinking about the most common form of lung cancer. Lung cancer is the most common form of cancer death in this country and around the world. What we've learned so far is that by looking at lung adenocarcinoma, the most common kind of lung cancer, we can see immediately that this disease can be subdivided by the most important cancer gene that's undergone mutation. So what's more, for many of these mutations, but not all, we have drugs that are partially or very effective. what you see at a glance is that we have about a quarter of these tumor types for which we have not yet identified the driver, about a third for which we have a driver but we don't have any drugs. I'll mention that a little bit later. And then quite a lot, almost 50%, very close to that, that have a mutation for which we have an available drug that's at least sometimes effective. So that's exciting, and it tells us how subcategorization can be incredibly informative and beneficial. But we also have to recognize that these tumors are very complex. And when you look at individual tumors across the horizontal axis and compare those with the names of different genes that are involved, what you see at a glance without decoding all the colors is that no single tumor has exactly the same pattern of mutations. That tells you that while there are some common mutations that obviously afford opportunities, that every tumor is fundamentally different. Just as every individual is different, the mutations that have occurred during life to produce that tumor are a different set of changes. Even more impressively, the number of mutations, if we examine the entire genome, which is now possible to do for just a couple of $1,000s, reveals that there are as many as thousands of mutations in certain kinds of cancers, especially when those cancers are caused by known carcinogens, such as exposure to UV sunlight or exposure to mutation-causing materials in cigarette smoke or in cooked meats. And in contrast, there may be only a few mutations in certain childhood cancers where the number of mutations may be less than 10. So this reflects, again, another amazing degree of variability and also allows us to begin to look in this constellation of mutations in any single cancer for patterns that might be revealing about what is responsible for those mutations. So just to take two simple examples, when we compare the kinds of mutations that occur, and you don't need to worry about what letters are being changed, you can see at a glance from these charts that on the left, the genetic signature, mutational signature in melanoma, a disease that is highly influenced by exposure to UV, is quite different from the profile that's seen in squamous cell lung cancer, where the major mutagenic or carcinogenic agent is tobacco. And there are now about 20-some signatures of this kind being seen, and I believe that those are going to be increasingly important in doing epidemiological studies to try to understand what kinds of exposures are conducive, pose risks for various kinds of cancer. Now, I've been talking about cancers as though this is a static situation. It's not. It's evolutionary. Many of you have probably seen this page from Charles Darwin's notebook in which he talks about a species labeled over on the left that undergoes continued variation and selection to generate the diversity of species that we see when we go out into nature. The situation is strikingly similar in cancers. Here's an example of a kidney cancer studied by another British man who works at Cancer Research UK, but the cancer begins with only a couple of mutations, but over the course of its growth and the production of metastatic growths at different sites in this patient, additional mutations have occurred. This creates a pattern such that when one looks at a tumor by sampling at different places in the tumor, you see the emergence of what we call subclones that result from continued mutation. And in some cases, it's possible to say those secondary mutations are of a particular type and even caused by certain kinds of changes in cell behavior or exposures to chemotherapeutic drugs or other mutation-causing agents. But the result is that when you sample this tumor at any single time, you see not just one cell type, you see a multiplicity that vary with respect to their mutational characteristics. And this obviously creates some difficulty with respect to diagnosis, with respect to making a choice of therapy, and with respect to telling a patient what the likely outcome might be. One of these secondary mutational types that is of particular concern are the mutational secondary events that make a tumor resistant to a chosen therapy. This constitutes the basis of what we call acquired resistance. A patient being treated for chronic myeloid leukemia with GLEEVEC sometimes will become resistant, the tumor will recur, and we'll look back and we'll see that patient's tumor has acquired another mutation which affects the target. So in acquired resistance, which is probably the biggest problem we're facing with the targeted therapies that are available today, we often find mutations in the target itself, which allows, for example, the enzymatic activity of the target to persist in the presence of the drug. That's a problem. What's making this an even greater problem is the resistance can be a lot more complex than that there are many ways that a cell can bypass the effects of the drug by turning on other cancer genes or by changing the way signals are transmitted within the cell. And we don't know all of those as yet. That's one of the big challenges being faced by those of us who are trying to carry out the precision medicine initiative. Another thing that's being changed dramatically are the way in which we do clinical trials. No longer do we group patients simply by the site at which the tumor arises, but we are increasingly grouping patients by the nature of the damaged genes in each individual tumor. And then choosing a therapy to be tested, not just because the tumor came from the esophagus or came from the lung, but choosing a therapy to be tested based on the alteration in specific genes and in the signaling pathways that are controlled by those genes. Furthermore, we're also now able to go back to previous clinical trials in which small numbers of patients did show a response to a drug and do a molecular analysis that tells us why that patient may have responded. And these so-called exceptional responders have revealed many new pieces of information that have encouraged drug development and encouraged testing of drugs in patients who have the mutations that were found in those exceptional responders. One example of this kind of approach is the massive new trial that the NCI is about to take on called the MATCH trial. And in this kind of trial, which exemplifies what we're trying to do in the Precision Medicine Initiative, we take in patients with any kind of tumor if the tumor has failed to respond to all known conventional therapies. We then carry out an analysis of the genes in that tumor, and we have a predetermined match of certain kinds of changes to drugs that have been made available through the generosity of drug companies that have been willing to engage in this adventure with us. That's been a very important part of the story. Well, what kind of thing do we hope to see? Obviously, we hope to get routine, good responses to some of the drugs that industry has developed, but the kind of thing that can happen and does happen is illustrated by this young man, Harrison, who's a 10-year-old boy who had one of the more common cancers of childhood, a B-cell or so-called acute lymphocytic leukemia. When he failed conventional therapy, so I mean by refractory, he was not put into a remission with conventional chemotherapy, which generally works in these kids. His cancer genome was carefully examined, and he was found to have an unusual deletion on one of his chromosomes that brought two genes together. One of those genes happened to be a gene that makes a protein, an enzyme, that the drug GLEEVEC, that I've already told you about, can work against. And as soon as he was placed on GLEEVEC, he went into a profound remission that's lasted for a couple of years. There he is with his oncologist touting the virtues of oncology. So these pictures simply show that his bone marrow returned to normal within weeks, that the bad cells that have the abnormal chromosome disappeared, and the bad protein, which can be seen initially, was no longer being made because all the cells that made that bad protein were being killed by the use of GLEEVEC. So this is the kind of really remarkable anecdote that gives us great hope that taking on trials of this kind is going to change the landscape of the way we practice oncology. Now, I'd be remiss if I didn't say something here about the immune system, because the immune system, which has long been thought to play an important role in governing the growth of tumors, has only recently been strongly validated as a means to control cancers. Those validations have come in many different forms through the use of antibodies that attack proteins on the surface of cancer cells, and you're all familiar with Herceptin, which has been doing that for over 15 years in breast cancer patients. That's been something not totally exceptional, but moderately exceptional. Through the use more recently of antibodies that actually have a poison attached to them, and that's been successful for some cancers, including some leukemias and mesothelioma. More recently, it's been possible to take one of the cells of the immune system, the so-called T cells that are actually the killer cells in controlling tumor growth, and to do a kind of genetic engineering, putting into those cells a device, an antenna that sees a property of the cancer cell and instructs the T cell to respond to it. And perhaps most impressively, there have been antibodies developed over the last decade or so that inhibit an unexpected aspect of the immune system. That is, the immune system has built into it a way to control excessive immune response. That makes sense. You don't want your own immune system responding to your normal cells. But this checkpoint system can be inhibited by using antibodies, and that's a remarkable thing. So it's been shown by Jim Allison and others that there are a set of molecules, now very carefully studied, that are the instruments by which immune blockade and the restriction on the immune system is carried out. And by making antibodies against those instruments, it's possible to block the checkpoint and unleash the immune system to attack cancers. And this is simply an illustration of how things are normally kept under control, but one can unleash T cells to attack cancers by using antibodies that attack the machinery that governs the so-called immune blockade. And there's a lot of new excitement about this because it turns out that while melanoma and maybe a few leukemias were the initial targets for this kind of therapy, there are now many successful applications of it in bladder, renal, lung, and other cancers. Moreover, this is going to be part of precision medicine because we are learning about what properties of tumor cells must be present for these responses to occur. And there's also an irony here that one is awed by the number of mutations sometimes seen in certain kinds of cancers, literally thousands, even tens of thousands of mutations. In this context, that's a good thing. It makes more foreign protein, or those thousands of mutations make more foreign proteins that the immune system is able to recognize. And I think all of us feel that it's very likely that the patients who are going to respond best, especially in these early days, to immunotherapy will be those who have many so-called passenger mutations that make new, novel proteins that can be recognized by the immune system, especially when the immune system is unleashed by these checkpoint inhibitors. So, can't tell you everything, but what remains to be done in large and sort of large letters, and what is the precision medicine money, assuming we get it from Congress, going to be used to do? Well, we haven't finished characterization of tumors just to build this catalog of information. Because tumors are heterogeneous, we need to start looking at single cells from tumors, not just a smoothie made from a tumor. We need better ways to monitor cancer growth and to look for cancers early, and we all believe that there are enough cells in the bloodstream and other fluids during the generation of a cancer and during the treatment of cancer to begin to use so-called liquid biopsies and molecular tools to follow cancers in that way. We still don't know how to do things like combine targeted therapies or combine targeted therapies with immune therapies and making better so-called preclinical models, cell lines, small collections of cells in culture, and cells that go into mice and grow in mice need to be improved so that we can begin to test how best to combine therapies and get the kinds of effects that are achieved in other contexts, like the treatment of microbial disease with combination therapies. And then we have cancers that have very common mutations, but they're mutations that we have not yet been able to address with new therapies. I've pointed out the frequency of RAS mutations in lung cancer. That's even more true in pancreatic cancer, where 95% or more of pancreatic adenocarcinomas have a mutation in the RAS gene. So we have started a big initiative up at our Frederick National Lab to try to address the difficulty of targeting cancers that have mutations in this difficult set of genes. We also need to be able to use computational methods more appropriately and powerfully to aggregate the information that's now available, interpret it, and disseminate it. And I remind you that in the original report on precision medicine, a key role is played by a knowledge network, a constellation of data that can be both enlarged by new clinical observations and new studies and used by practitioners of oncology who are treating patients. And we need to build those knowledge networks, and that's the NCI has been dedicated to doing that. We are building a so-called genomic data commons and testing out new ways to use cloud computation to make this data much more manageable. And the general notion is to build a cancer knowledge system in which every cancer patient considers themselves to be a cancer information donor, building on the altruism that we see so often in cancer patients. Attempt to get information about individual cancers using the methods I've talked about and pull all that together to make changes in the way we treat patients, and then use the consequence of those new treatments to build yet a deeper set of information about how cancer responds. My final point is to point out that we need to address some practicalities. The transition from what we do now to what we'll be doing in the future is going to challenge the economics of healthcare and the regulatory apparatus that we use and the way in which patients get involved as information donors. And just give you a couple of examples of how people are thinking about this. First, we've got to choose appropriate genetic tests, and they have to be appropriately overseen and regulated, in my view. But we have a lot of choices now. You can take a patient's tumor, you can sequence the whole genome, or you can sequence a part of it, or you can look at the genes that are being expressed, or you can look at just a few genes. The costs go down, the information also goes down, and there's an active debate about what should be done to characterize the tumor of individual patients now, given the therapeutic options and given available resources. The resources, the financial resources, need to be contemplated or thought about in the context of what the consequences of not taking these next steps might be. The French have done a very good job in trying to make some specific assessments when a drug became available for one specific kind of lung cancer. They did an economic experiment in which they followed everybody in France who was diagnosed with this form of lung cancer, adenocarcinoma, the most common form, was subjected to testing for mutations in a gene for which the drug was suitable. And the testing itself was relatively inexpensive, just a little under 2 million euros, and that identified about 10% of the patients, 12% of the patients who had the mutation, needed the drug. They got the drug. The cost of the drug was was clearly high, but you spared 15,000 patients who tested negative from getting that drug, and you were allowed them to get on more appropriate therapy because they weren't going to benefit from the drug. So this all makes sense economically, and that kind of analysis needs to be done for many other targeted therapies as they come along. Finally, we need to think about how this is going to be done in practice, and we are already seeing a pattern in which patients are going to be treated as they were initially, with a microscopic evaluation and imaging, and they will then be looked at genetically to determine which drug might be the appropriate drug for the mutation that's found in the analysis of their tumor. and getting this to be a streamlined process in which doctors who are not trained in molecular biology can make good use of this information will be a major challenge for us. So that is the story. So I've told you something about the big picture and what we're trying to do with the President's new initiative, and as we try to continue this battle against cancer that involves understanding cancer better, learning how to diagnose it more appropriately, treat it, and prevent it in a more rational fashion. I'd be happy to take questions. Thank you. Yes, ma'am.
Yeah, that really colorful slide that looked like Legos, that was what, the single mutations.
Of the signatures?
Yeah. I didn't read it fast enough, but it looked like there's like two bases that change, like C's become something else.
Well,
I mean, did the other two never change or something?
no, the point, that's a good question. So she's asking why you see certain kinds of changes in certain situations. That was the situation in melanoma, which is to a very large extent attributable to mutations induced by ultraviolet light. We have known for many years that ultraviolet light causes a specific change. A C gets changed to a T. So that's why you saw so many of that kind. If you look at other signatures, you'll see other kinds of changes that are more characteristic, for example, of exposure to a chemotherapeutic drug that makes A to G changes. So you've got the key point. Yeah.
Thank you, Doctor. Can you discuss the timeline a bit more? If there's an exciting breakthrough discoveries like the Nature magazine covering us, or others who talked about, how long would it take for something like that to directly reach a larger group of patients? The knowledge gained into development of treatments and medicine, and what can be done to accelerate that, so it's not just research and trials and all of that?
Well, I would not use the word just, but I think that question is too case specific. In other words, give you an example. My own laboratory is generally interested in lung cancer, and we've gotten more engaged in studying lung cancers over the last 12 years or so because when I was at Sloan Kettering, we noticed that many patients with lung cancer who received a drug that had already been developed as an inhibitor of a certain kind of enzyme was very effective in producing remissions in about 10% of patients. So we and actually two other groups up at Harvard started looking at these cancers for mutations in the genes that might be affected by that drug. And they were found. And immediately, within, at least at our center and other centers around the country, and now virtually everywhere, patients are tested for that mutation and that drug is used. Another example, a few years ago, another form of lung cancer was found to have a mutation in a gene that had been discovered in a leukemia as a mutant gene. And there was already a drug to treat the leukemia, and that drug was approved for treatment of the lung cancers that had this specific novel mutation within a matter of a year or so. But finding a mutation in a new gene, which we frequently find through the Cancer Genome Atlas project, may require, unless we're lucky, a large amount of basic science before we can begin to think about how to develop a drug against that kind of mutation. So a few years to many years. And I think it's also important to remember that what you see as a big breakthrough may depend on something that happened 20 or 30 years ago. We'd be nowhere without the basic principles of recombinant DNA technology and means to sequence genomes and analyze cells for their properties in carrying out certain processes in response to signals they receive from the outside. All these forms of science are now focused on trying to solve some of these clinical problems, but we can't ask the question just in the context of getting from what you might think is a breakthrough because you can understand how it might be applied. One of the things that I am concerned about, and many other the leaders of the NIH are concerned about, is that in this very constricted environment for funding where there are so many opportunities to do things that are on the clinical edge, we don't want to lose the possibility of making fundamental discoveries. We know very little about how life really works. We have some very important things that have been discovered, and it allows us to get to the point that we're at now, which is a very exciting point. But we all know from things that have happened over the last year or two that allow, for example, much more effective manipulation of whole genomes to study the way in which mutations affect cells. Those things would not have been found if there was not a cohort of investigators who was not thinking about curing somebody, but just trying to understand the fundamentals of life.
There's been talk about personalized medicine for some time. What's new and different about this from precision medicine?
Personalized and precision medicine are basically the same thing, but I don't like the word personalized because my father practiced personalized medicine. He knew his patients. We're talking about something that has to do with precision, and there is an individual property here. And I don't, I know what people mean when they say personalized medicine, but it does, it's a fuzzier term, and I don't like euphemisms.
Where's the money coming from?
Where's what?
Where's the money coming from for the precision medicine?
Well, it will ultimately coming from Congress, but the president's proposed an increase for the NCI of $70 million in the next year's budget. These are things we wanted. That's against a base budget of almost $5 billion. So the program, we could spend a lot more, and we do spend quite a lot on these things. What I showed you was not what we proposed to do. It's what's been done over the last several years, and that's been done with the resources of the National Cancer Institute and actually other institutes and other funding agencies outside the government. So there are a lot of sources right now. The biggest source for this national and international effort comes from the NIH and other major funding agencies. Some comes from the drug companies, obviously, that are making drugs that can be used to treat cancers based on these mutational events.
Going along with the drug companies, and where does that collaboration start? Or is it a case-by-case basis? Is there usually during preclinical trials? Is it more like, do they come to you or you go to them?
It's a case-by-case. It's a very good question. And in the case that where I happen to mention the drug companies specifically, the so-called match trials, we've gone to the companies. We've said, here's what we propose to do. There are going to be thousands of patients enrolled. You guys all have an investment here. You're making targeted drugs because you know that they are effective, they're relatively non-toxic, you can sell them for very high prices, and that's not an insignificant factor. And we can help you with the testing because we're going to be looking at thousands of patients, and we're going to find the patients who are most likely to respond. Let me give you an example. 12 years ago or so, when we started working on lung cancer, there was a drug that I actually alluded to, two drugs that attacked the kind of mutation we were studying. One company was testing a drug of that type against all patients with that kind of lung cancer. And their trial failed because there weren't enough patients in the trial who had the mutation. So design of trials is extremely important in this context, and for us to do the genetic part, provide the basic knowledge, them to provide drugs is a pretty good deal. And we have a national clinical trials network that is lining up all these patients, and there are thousands of clinical investigators around the country recruiting patients into these trials. So we provide benefit, and they provide some drug. Yeah, but there are other ways it works too, but it's a two-way street. Yeah.
So, sort of following up on that line of question, so a patient database, does drug companies have access to patient data? What are the, from a practical matter, how do you, I guess, protect people's personal information, and then who does have access to the database?
Yeah, well, that's an important question. We obviously believe in data access, but there is controlled access for most of this information at the moment. We depend very heavily, and I think everybody must in the future, on consent. And I find that when I talk to patient advocacy groups, that they just want science to advance. They want information to be used to advance our science. And most cancer patients are not so concerned about being identified through the use of this kind of data. Nevertheless, I do think that there are some people who are concerned, and it's not that difficult to create a new kind of consent form in which people can agree to different levels of exposure. That is, It's possible for us to retain information and give it only to certain people who are qualified users, is one example of how we can control the information.
So is that what's occurring, or is that what
That's
you're
largely what's
suggesting?
occurring now, right.
So
It's
who does
controlled
have access to the NIH database then?
People who we say are qualified.
So NIH is administrator for that.
Yeah.
What do we know about, at this moment, about cancer and epigenetics? In other words, I've always thought about this. What happens if, the person A, their grandparents worked in a coal mine or something, and then, can they pass down?
We're talking about something, the term epigenetics, just to clarify, means an event that affects the way in which DNA functions, as opposed to changing the DNA in a structural way. So in that sense, epigenetics is emerging as extremely important. Transmission of a mutation from a parent to a child is extremely unlikely based on environmental exposures that are occupational.
Can the expression patterns for which genes turn on and off, that can be...
In principle, that can be transmitted from one cell to daughter cells. Whether it actually be transmitted to the germline is a more difficult question. There are some examples that suggest that may be possible in some circumstances. But that, to my mind, is not a major feature of dealing with cancer right now.
Very educational presentation, thank you. But I was wondering, there was an episode of Vice this season. I don't know if you watch Vice on HBO. No, I don't. It's like a documentary news cast where they were this season. One of the episodes consisted of treating cancer with like measles or different diseases, and I was wondering.
So there is interest in the possibility of using viruses to attack cancer. Using viruses to attack cancer cells. I would say the jury is out. There are different viruses being tested. I would watch with interest. There are risks, obviously, in using a virus that is infecting cells to try to cure cancer, because yes, in some cases, but how reliable that is, after all, as you learned today, cancers are very heterogeneous. How universal that's going to be, it's a risky business to use a live virus that's also a pathogen to treat a cancer. But there's studies going on that NIH is supporting to see whether there may be any prospect of developing treatments in that way. And obviously, we know enough about how to manipulate viruses to try to change those viruses so they're less pathogenic to normal cells and may be targeted to cancer cells. So there are lots of aspects of what's so-called virolytic therapy that are being studied.
I know there's some things that we know, and there's a whole bunch of things that we don't know yet. And my question relates, I guess.
That's true about everything.
Yeah, to the French study that you were talking about.
The economic study?
Yeah. And I'm thinking about that article in the Post a couple weeks ago about the oncologist, that the woman was not responding to colon cancer chemo, and so he tested her tumor and gave her a breast cancer chemo, I think it was, and it worked. So now that we know that, why can't we just do that? I mean, the things that we know, why can't we just do them?
Well, we should be able to do it. And that actually, one of my goals when I go to New York is to make this information available to ensure that everybody in that region is aware of these advances. Unfortunately, not everybody keeps up with everything that's happening in every field. So there are people who are not getting drugs because their DNAs have not been their tumor DNA has not been properly tested, and because the healthcare provider doesn't know about some of the latest things that have happened.
But if everybody knew it, could they just do it, or is it like, oh, the insurance companies say it's too expensive, or?
No, insurance companies cover these therapies.
So people just need to know to look for that, and they can look for it.
Correct.
Great, thank you.
Yes, ma'am.
Thank you, Dr. Varmus. I have a question about the 1 million-person cohort you guys are planning to enroll. I haven't quite figured out from what I've been reading whether you're planning on enrolling individuals who have no preset.
I would suggest that you invite Francis Collins to come and talk to the caucus. But to answer your question in a more straightforward way, I'm not myself. My part of the precision medicine initiative is what I talked to you about today. There is a committee that has been assembled, outside scientists, to try to determine what the cohort study is going to be. And that group, there was a workshop held a couple of weeks ago you may have heard about. I would say no clear decisions have been reached, but there are other cohorts that exist that might be assembled. I think it's also important not just to think about who's going to be in the cohort, but what questions are going to be asked, what kinds of tests are going to be done. Sure.
What I wanted to ask is, how would you do it if you were sticking around instead of, unfortunately?
Well, you know, that would not be my part of this initiative. Nevertheless, we have a lot of cohorts at the NCI, and those cohorts are assembled to answer specific questions. For example, some of our most famous cohorts are cohorts assembled of pediatric patients who've had cancer. Pediatric cancer is a common, perhaps the most common cause of death among kids outside of accidents, but it's still a fairly rare disease, happily. But we have large cohorts of individuals, of kids who have gone through cancer treatment and survived, and it's extremely important for us to be following those kids. In fact, we now know about losses in intellectual capacity, about second cancers, many things we learned in trying to understand the secondary aspects of apparently successful treatment of lung cancer, keeping a kid from dying. Doesn't mean the kid has gotten a fair break in life, because they're often impaired and they're much more likely than their peers to develop secondary cancers. And only cohort studies that are designed to answer specific questions will be able to do that efficiently.
There's a placeholder language for precision medicine in the 21st century.
Yeah, I noticed that.
What types of things would you like included?
Well, I'm not sure we need additional language. You know, we've been given an imprimatur to use our skills as scientists to learn more about cancers, to develop better ways to treat and prevent them, and give us the money. I don't need language.
Yeah, will there be, or has there been an emphasis on studying the potential genetic differences between persons of different races or of different ethnic origins?
Yeah, absolutely. I mean, obviously this is a complicated question because, an African American population is very complicated. It's not one and ditto for any other ethnic grouping. It's often self-declared, and races are mixtures, and so these are tough questions. But I would say, I'd make a more general statement that we haven't yet done as good a job as we should in learning what these acquired mutations, so-called somatic mutations, mean in the context of different genomes. You and I share 99.9% of our genomes, but that extra 1 in 1,000 makes a big difference, because that's literally millions of differences, because our genomes are big, 3 billion base pairs. So having a better understanding of how a mutation, identical mutation in your genome and my genome would behave in the context of the differences we inherit is really critical. So it's a much broader question than you're asking, but we are paying attention to the fact that there are important differences between Hispanics and blacks and whites, and we need to take that into consideration to be sure we're getting not just a good ethnic mix, but a good genetic mix. One of the limitations of studying cancer in mice is they're all the mice in an inbred strain are all the same. That's advantageous experimentally, but in the real world, you miss learning some of the things we need to learn. So having highly diverse populations is one way to try to ensure that you'll have a lot of genetic mix in the background. Other questions? If not, I'm glad to see there are at least a few bags left in the back, and I'm going to grab one and go back to NIH. Thank you very much.